This hominid operates without a cell nucleus. Instead of derives its genetic expression, its physiology uses a novel fluid transport. True neuromorphic networks replace standard neurons.
For 4 billion years, biology has been authored in the singular language of DNA. But this is not an inevitable law, but rather a frozen accident of Earth's early chemistry. In the RNA world, genetic continuity was maintained by self-replicating molecules. These strands could both store information and catalyze reactions. Primitive lifelike systems could have originated through complex, self-sustaining metabolic networks driven by thermodynamic gradients, without relying on an initial genetic polymer.
Driven by geothermal gradients, autocatalytic networks spark metabolism, self-replicating polymers emerge, diverging into vulnerable RNA and resistant XNA. We are exploring alternative biochemical frameworks, synthetic polymers known as XNAs. They replace the standard furanose sugar backbone. Because natural polymerases and nucleases cannot recognize or process these non-natural backbones, organisms establishing an exceptionally biochemical foundation.
To support a multicellular hominid devoid of DNA, alternative polymers like PNA and TNA are required. These systems enable high-fidelity replication and ensure full epigenetic continuity. Peptide nucleic acid replaces the sugar phosphate backbone. Its neutral backbone eliminates electrostatic repulsion. This allows for strand invasion. It binds with exceptional stability.
This high structural stability prevents degradation by endogenous nucleases or proteases, allowing PNA templates to persist within the cellular environment without continuous enzymatic turnover.
Threose nucleic acid, or TNA, is a synthetic genetic polymer. It features a backbone 1 carbon shorter than natural DNA, yet it forms stable duplexes, pairing with DNA and RNA. The structural simplicity of TNA relative to ribose makes it a strong progenitor. Specialized polymerases enable the enzymatic synthesis of TNA. Furthermore, the synthesis of TNA, with an expanded genetic alphabet containing unnatural hydrophobic base pairs, such as the TPT3, allowing for the encoding of novel biochemical pathways. DNA uses deoxyribose. RNA uses ribose. PNA has peptide linkages. TNA is based on threose.
DNA and RNA have negative backbone charges. This governs their structure and interactions. PNA is neutral, unlike most nucleic acids. TNA returns to a negatively charged backbone.
Stability and is susceptible to DNases, with minimal stability against RNases. PNA provides high resistance to nucleases. TNA, resisting serum nucleases. Chemical modifications to histone tails modulate chromatin. Acetylation and methylation local gene expression. HP1A bind to these methylated sites. This drives the spreading and inheritance of silent states. Complex populations of non-coding RNAs regulate target transcripts post-transcriptionally, forming feedback loops that maintain stable cellular phenotypes.
The human genome is dynamic, not static. Mobile elements actively shape our DNA. This process, called retrotransposition, creates a somatic mosaic.
Furthermore, proteinaceous inheritance can bypass nucleic acid templates entirely through the autocatalytic propagation of amyloid conformation. Prion proteins propagate by transmitting a highly stable structure. This conversion occurs through nucleated polymerization, where existing amyloid fibrils grow at their form ends. Templating allows structurally distinct populations to form, enabling variation and adaptation. The normal prion protein, PRPC, can spontaneously misfold or be induced by a pathogenic seed. This leads to amyloid fibril growth.
This hominid operates without somatic DNA. Highly specialized tissues minimize mechanical wear. Enucleated cells rely on pre-synthesized proteins, maintaining viability without transcription.
These cells undergo programmed enucleation. This establishes a durable physical shield. It protects the viable tissues underneath.
When ancient DNA is lost, researchers trace human evolution using stable sugars preserved in fossils. This shift provided our ancestors with ancient malaria.
In fossils devoid of DNA, preserved biochemical footprints serve as a reliable molecular timestamp to trace hominid evolution directly.
To maintain a human-scale body size without continuous renewal, these organs utilize specialized physical barriers.
The speculative plan features muscular valves. Abdominal pumping drives air through the trunks. Tracheoles bridge the aqueous diffusion gap. Tubules manage dry uric acid excretion. The fragile mammalian alveoli are completely replaced. A branched tracheal system delivers gases directly to somatic cells. This bypasses the need for blood-mediated hemoglobin transport. Biophysical modeling reveals a transport asymmetry. Oxygen is constrained by the thin aqueous tissue gap. Fick's law defines this diffusive flux. Carbon dioxide is limited by tracheal geometry. Tracheal tubes limit carbon dioxide transport. Fick's law defines this diffusive flux to oxygen, but high solubility allows CO2 to diffuse rapidly across tissue. This high solubility makes the geometry and ventilation of the air-filled tracheal tubes the primary limit. Conduits are structurally reinforced with spiral chitinous rings, preventing air collapse. Insects actively pump their abdomens. This clears out stagnant air. It keeps surfaces oxygen-rich. This drives carbon dioxide out. The foregut is protected by a rigid chitinous lining. In the midgut, a peristrophic membrane filters nutrients. The hindgut's thin intima permits water recovery. The midgut lacks a rigid chitinous lining to permit nutrient absorption. It is protected by a semi-permeable peristrophic membrane. It acts as a filter, allowing enzymes and nutrients to pass.
To assist digestion, the stomach lining is colonized by bacteria. These microbes specialize early. They digest tough fibers, producing fatty acids absorbed across the gut wall.
As this fluid flows into the rectum, the complex wraps the Malpighian tubules closely against the rectal wall. This creates a concentrated environment that extracts water from the fecal pellet. Lacking nuclear DNA, these cells utilize soft semiconductors. They transport both electronic currents and ionic fluxes, completely bypassing traditional nerve conduction.
Applying a gate voltage injects ions. This changes its doping state and conductivity. This mechanism mimics voltage-gated channels. It works without any protein-based structures.
These OECNs use vertical architectures. Spikes match biological nerve cells, traveling along myelinated axons via saltatory conduction. Glial cells regulate homeostasis. This active buffer system prevents runaway extracellular potassium accumulation, stabilizing signal conduction and ensuring prompt delivery of sensory information to the brain.
The dense network acts as the primary sensory system, converting physical loads into signaling cascades.
Bone matrix generates localized electric fields. Shearing forces cause collagen fibers to slip, breaking inversion symmetry, generating a potential. This equals 1/10 the constant of quartz. When bone is compressed, hydroxide crystals deform. This mechanical stress triggers internal polarization. Resulting surface charges bond with collagen fibers. This process prevents structural weakening. Compressive force on the bone matrix triggers a reorganization of dipole moments. This electrical signal drives calcium entry through CAV and PAEK precursors, enhancing bone formation.
Simultaneously, mechanical shear stress triggers the secretion of nitric oxide and increases the expression. OPG inhibits osteoclasts and decreases bone resorption. In a hominid lacking somatic DNA, this direct electromechanical coupling allows bone structure and mineral density to be maintained dynamically through physical activity. In organisms with enucleate cells, regeneration relies on reprogramming. Macrophages trigger this change, reverting cells to a stem-like state.
Macrophages do more than just clear cellular debris. These cells become local without mitotic division, sustaining lifelong tissue repair and homeostasis. Synthetic biology builds artificial cells. RNA origami creates a cytoskeleton, providing mechanical stability and shape control. These systems operate without DNA.
Behold the blueprint contrasting human and post-biological systems. Canonical human organisms rely on DNA replication. Post-biological hominids utilize deformed protein templating.
Let us contrast the visceral barrier of both systems. Canonical humans rely on self-renewing mucosal cells. Post-biologicals use a dense chitinous intima. This provides pure mechanical protection.
Let's examine the comparative blueprint. Canonical skeletal modeling is receptor-mediated.
Post-biological matrix-induced stress gives way to piezoelectricity. Enucleate hominids demonstrate life without DNA. Stable synthetic polymers complexity. Electrochemical networks mimic nervous systems. These systems provide a cohesive biophysical model.
Kira, Kira, Sahab Sama! Mirror, mirror on the wall, VIPs are standing tall. Shiny suit and power face, ruling over time and space.